What is biochemistry of proteins? Maayong adlaw, I am Agalin Kai, a student of Norso, and we will be talking about the general concept, the biochemistry of proteins. A protein is a naturally occurring extremely complex substance that consists of amino acid residues joined by peptide bonds. Proteins are present in all living organisms and include many essential biological compounds such as enzymes, hormones, and antibodies. Where does protein synthesis occurs? Protein synthesis occurs in the ribosomes of cells. In eukaryotic cells, ribosomes are found as free-floating particles within cells and are also embedded in the rough endoplasmic reticulum, a cell organelle. What does proteins do? Proteins are essential for life and are essential for a wide range of cellular activities. Protein enzymes catalyze the vast majority of chemical reactions that occur in the cell. Proteins provide many of the structural elements of a cell, and they help to bind cells together into tissues. Proteins in the form of antibodies protect animals from disease, and many hormones are proteins. Proteins control the activity of genes and regulate gene expression. More about it. Protein, highly complex substance that is present in all living organisms. Proteins are of great nutritional value and are directly involved in the chemical processes essential for life. The importance of proteins was recognized by chemists in the early 19th century, including Swedish chemist Johannes Jacob Berzelius, who in 1838 coined the term protein, a word derived from the Greek proteus, meaning holding first place. Proteins are species-specific, that is, the protein of one species differ from those of another species. They are also organ-specific. For instance, within a single organism's muscle, proteins differ from those of the brain and liver. Proteins are polypeptide structures consisting of one or more long chains of amino acid residues. They carry out a wide variety of organisms' functions, including DNA, replication, transporting molecules, catalyzing metabolic reactions, and providing structural support to cells. Let's go deeper with the four structures of protein. A single protein molecule may contain one or more of the protein structure types: primary, secondary, tertiary, and quaternary structure. Primary structure. Levels of structural organization in proteins. Primary structure, analytical and synthetic procedures reveal only the primary structure of the proteins, that is, the amino acid sequence of the peptide chains. They do not reveal information about the conformation of the peptide chain, that is, whether the peptide chain is present as a long straight thread or is irregularly coiled and folded into a globule. The configuration or conformation of a protein is determined by mutual attraction or repulsion of polar or nonpolar groups in the side chains of the amino acids. The former have positive or negative charges in the other side chains. The latter repel water but attract each other. Some parts of a peptide chain containing 100 to 200 amino acids may form a loop or a helix. Others may be straight or from irregular coils. The terms secondary, tertiary, and quaternary structure are frequently applied to the configuration of the peptide chain of a protein. A nomenclature committee of the International Union of Biochemistry has defined these terms as follows. The primary structure of a protein is determined by its amino acid sequence without any regard for the arrangement of the peptide chain in a space. The secondary structure is determined by the spatial arrangement of the main peptide chain, without any regard for the conformation of side chains or other segments of the main chain. The tertiary structure is determined by both the side chains and other adjacent segments of the main chain without regard for neighboring peptide chains. Finally, the term quaternary structure is used for the arrangement of identical or different subunits of a large protein in which each subunit is a separate peptide chain. Secondary structure. The nitrogen and carbon atoms of a peptide chain cannot lie on a straight line because of the magnitude of the bond angles between adjacent atoms of the chain. The bond angle is about 110 degrees Celsius. Each of the nitrogen and carbon atoms can rotate to a certain extent, however, so that the chain has a limited flexibility because all of the amino acids except glycine are asymmetric L-amino acids. The peptide chain tends to assume an asymmetric helical shape. Some of the fibrous proteins consist of elongated helices around a straight screw axis. Such structural features result from properties common to all peptide chains. The product of their effects is the secondary structure of the protein. Tertiary structure. The tertiary structure is product of the interaction between the side chains of the amino acids composing the protein. Some of them contain positively or negatively charged groups. Others are polar and still others are nonpolar. The number of carbon atoms in the side chain varies from 0 in glycine to 9 in tryptophan. Positively and negatively charged side chains have the tendency to attract each other. Side chains with identical charges repel each other. The bonds formed by the forces between the negatively charged side chains of aspartic or glutamic acid in the one hand and the positively charged side chains of lysine or arginine of the other hand are called salt bridges. Mutual attraction of adjacent peptide chains also results from the formation of numerous hydrogen bands. Hydrogen bands form as a result of attraction between the nitrogen bound hydrogen atom and the unshared pair of electrons of the oxygen atom in a double-bonded carbon-oxygen group. The result is slight displacement of the imide hydrogen toward the oxygen atom of the carbonyl group, although the hydrogen bond is much weaker than a covalent bond. So, the type of bond between two carbon atoms which equally share the pair of bonding electrons between them. The large number of imide and carbonyl groups in peptide chains results in the formation of numerous hydrogen bonds. Another type of attraction is that between nonpolar side chains of valine, leucine, and isoleucine, and phenylalanine. The attraction results in the displacement of water molecules and is called hydrophobic interaction. In proteins rich in cysteine, the conformation of the peptide chain is determined to a considerable extent by the disulfide bonds of cysteine. Thus, the halves of cysteine may be located in different parts of the peptide chain and thus may form a loop closed by the disulfide bond. If the disulfide bond is reduced, hydrogen is added to two sulfhydryl groups. The tertiary structure of the protein undergoes a drastic change. Closed loops are broken and adjacent disulfide bonded peptide chains separate. Quaternary structure. The nature of quaternary structure is demonstrated by the structure of hemoglobin. Each molecule of human hemoglobin consists of 4 peptide chains. The 4 subunits are linked to each other by hydrogen bonds and hydrophobic interaction. Because the 4 subunits are so closely linked, the hemoglobin tetramer is called a molecule, even though no covalent bonds occur between the peptide chains of the 4 subunits. In other proteins, the subunits are bound to each other by covalent bond. So, that's it for the biochemistry of proteins. This is Agalin and see you on the next video.